Hi, my name is Maggie. I'm a part of this year's iGEM team at the University of Oregon. We're headed to the night campus, where this year's team is focused on researching how to create a novel probiotic to treat bacterial infections in the GI tract. Stay tuned. Hey Ari, what inspired you to join iGEM?
I think what inspired me to join iGEM is the challenge, one. Two, the opportunity to work on something of my own, designing something and working through something and solving problems using new knowledge, old knowledge, yeah.
I hadn't yet had the opportunity to join one of the regular labs on campus, and I just really wanted the opportunity to grow as an independent researcher.
So I heard about iGEM from my friend last year, and I'm so excited to work with a lot of people.
I was inspired to join iGEM to learn more about synthetic biology and have a very hands-on experience in the lovely night campus.
What's our project this year, Emily?
Let me think. This year we are designing a probiotic strain of E. coli that can target and fight pathogens in the GI tract. One part of the project involves conjugating a toxin from our strain of E. coli Nissl into the pathogen.
Oh look, there's Luca. What's this year's project all about?
So this year's project is focused on developing a modular platform wherein a engineered probiotic strain of E. coli targets and kills pathogenic strains of E. coli. We want to change out that pathogenic bacterial strain to anything that we'd want to target. This year, we're testing our system against a pathogenic strain of E. coli called enterohemorrhagic E. coli, or EHEC for short. We're essentially trying to use their own technology against them, but they don't know that it's technology because they're bacteria. Namely, that's conjugation. And conjugation is a type of horizontal gene transfer. So we're literally trying to engineer our probiotic, a strain of E. coli called Nissl 1917, which is deemed as safe by the FDA and as widely recognized as a probiotic, to transfer a DNA encoded toxin to a pathogenic strain of E. coli and kill it, yeah.
What does that entail in wet lab?
Right now we're doing a lot of experimental design, so before we get into the wet lab, we kind of have to put the pieces together. So we have several different parts. We have the conjugation machinery. We have specificity that we need to bring into our system, which we're doing using fusion proteins. And then we also have computation crossover to the wet lab further down the line if our plans go well.
Hi Bella.
Hello.
What part of our project are you involved in?
I'm currently working in the wet lab to develop our modular conjugation system, and I am also on the entrepreneurship team working to improve our fundraising, and the education team to make fun educational videos for students of all ages.
I'm working on a lot of parts, making our conjugation system, making our killing system, our cargo. We're also working on a model, computational model for determining the binding affinity of antibodies, of nanobodies specifically.
I'm doing most of the computational stuff. How do I explain this simply? We are trying to build a machine learning model to predict and optimize the nanobody that bind to pathogens.
What's one thing you've learned so far?
One thing I've learned so far is that you have to develop a wealth of knowledge in order to engineer something. There's so many things that you can learn from engineering, and there's so many things that you have to learn in order to actually put parts together that work. And so working with other people is very fun.
Definitely teamwork, even though we're such a small team. In iGEM, we get minimum guidance from our mentor and our PI, so we get a chance to engineer things independently.
The biggest thing that I've learned has been how to integrate creativity when approaching and designing novel biological systems. Engineering is a field that takes a lot of inherent creativity.
Ooh, I've learned a lot, but I would say something that's really important is how to properly use a pipette.
Why should people donate to iGEM?
People should donate to iGEM because we need money to do science, to put it frankly. There's a lot of technology and materials and reagents that we need to perform the experiments that are necessary to get results. Another big piece of iGEM is attending the Paris Jamboree, where we can present our research, get amazing exposure to the biotech industry and meet the leads in those fields, as well as develop our science communication skills, refine our interests within synthetic biology. And all of that stuff takes money to get to happen. And we're a lovely group of students, so you should fund us.
Thanks, Luca.
Your funds will directly go to our lab supplies to help us continue our research and have a larger impact in the realm of infectious disease research and synthetic biology, and will also go to funding members of the team so that they're able to attend the iGEM Jamboree this year in Paris.
Because science is not strictly about being in the wet lab. It's not about working at the bench the whole time and your pipetting skills. It's also about the way you communicate your science and the people you talk to, the networks you build, and the resources you can kind of procure. We are a group of very engaged and driven individuals who are trying to advance their science skills and communicate good science. So if you donate to our cause, then we can do more science, we can make more of an impact, we can build a bigger network, and we can get more people involved at UO as well.
This year we're really aiming for gold, and we're doing a lot of really awesome research this year, so we're hoping to get some awards.
Yeah, go Emily.
Thank you for your support.